Aswin is using Malay. Okay, so transpirasi dalam tubuhan, transpiration in plant. So before we start, we comes out with the experiment, one of the experiment first. So tujuan or aim, membuat pemerhatian untuk mengkaji proses transpirasi dalam tubuhan. Okay, so observation to study the transpiration process in plants. So material atau bahan, we have pokok berpasu, bag plastik lucina, transparent, dan kertas cobalt chlorida kontang. Okay, potted plant, transparent. Wait, Okay, so I think we can use back English because somehow he choose to join the sejarah on the, I think. So now we have a transparent plastic bag and anhydrous cobalt corrida paper. Then we prepare the setup like this. So we stick 2 pieces of this anhydrous cobalt chloride paper in the transparent bag, and then we wrap it on the plant. Make sure that the plastic bag must be fully dry lah. Okay, sorry. If it's not, maybe there's gonna have some how to say, because you see the main idea here, I think is something, because this cobalt chloride paper, they are using it to test whether there is water or not. So if the plastic bag is with water, it's not dry, so it will change the color. So your result might be not accurate. Okay, then, all right, now we just continue. Okay, then we wrap it. Okay, and then the tree, we left it for 2 hours outside the laboratory, and then we observe what happened. So you will find out that there will be some liquid droplet form in the inner wall of the plastic bag, and your anhydrous cobalt chloride will change color from blue to pink. Means that there's water. Okay, so let's see. So okay, so what's the functions of the cobalt chloride paper in the experiment? This is first to test for the presence of water. Wait, I think I had a message coming. Okay, so then so this is why the functions of cobalt chloride paper. All right, so the inference, okay, inference of this observation. First, the liquid droplet are formed on the inner wall of plastic bag because why? Because the plants carry out transpiration. And the color of the anhydrous cobalt chloride paper change from blue to pink. Why? Because the droplet is water. If there is a existence of water, so the anhydrous cobalt chloride paper will change the color. Okay. All right. Next. So from here, let's check what's transpiration. So for transpiration, is the process of losing water, okay, in the forms of water vapor by evaporation through the stoma. Okay. So normally it happens during daytime. Okay. So transpiration will reduce the water contained in the plant. So that's why the amount of water in plant become less than the soil. And that's why cost less than the soil. And this will cause the water from the soil to be absorbed by the root because of the, you remember last time I mentioned about the difference in concentration will force the water movement. Remember, the water will move from low concentration area to high concentration area. Okay, remember? This is how it's trying to play, because when the water is lost, okay, when there's a water loss in plant, the concentration of plant inside the plant will become higher, correct? So less water. So the water from the soil will move, the water from the soil will move into the plant to balance out the concentration. Okay? Wait. Okay, so let's see. So this is how okay, the motion of the, okay, the movement of the water, okay, during the, what you call that, so it's from the soil, it moves from soil to the root, and then to the root through here, and then from here go to the leaves, and from the leaves it goes up. Okay, so this is how the water movement. Okay, I think we better use this one because this one seems like a bit easier. I think if you want to do note or anything, correct? You like this one more or the old one? Hmm? You're okay with anything. All right. So now, so let's see. Okay, the other role of transpiration. So the role of transpiration. Okay, not just getting rid of excess water. So it also transport the water and dissolve material from root to whole plant. Okay, so the other way plant to remove water, yes, there's a way. So nighttime, the plant will remove the excess water through the process of guttation. Okay, the how is it is the water is lost in the form of liquid through the pore we call hydrator found at the tip of the vein leaves. So normally, okay, this is where you see the vein at the tip here. Normally, this is where the droplet of water comes in. Okay, in Chinese, you know, in last time, all those wealthy men, they will say they collect the water from the flowers, to cook the tea. You know, those water is actually this guttation water. Okay, can why they say the water there's some aroma inside or there are some taste inside because there's a mineral, there's a mineral. There's mineral inside the water, the guttation water. So that's why when they cook it for the tea might have different aroma. Okay. All right. So in conclusion, so the transpiration somehow is a process of losing water in the form of water vapor by evaporation through stoma. So this is the one. All right, now let's see the factors that affect the factors that will affect the rate of transpiration. So let me see whether I can just straight away. I don't want to show this. I mean that I straight away use the theory. Is it possible? Because somehow there should be some theory only. Oh no, they are using the form. They are using the what you call that. All right, the first one is the light intensity. So I just jump out. So the higher the intensity of light, the higher the rate of transpiration. Okay, so I just we just go through the theory. Okay. It didn't show up. Wait, I'm trying to. Okay, you see how they do the experiment. You see where they control the light intensity. Okay, and then putting this set up the experiment like this, and then they put this inside a different condition under light, low light, or dark. Okay, so what happened here? You can see the when the presence of light is higher. Okay, when there is a presence of light, you can see the transpiration rate will be higher. Okay, so this is like what I said just now, like upside. So for humidity, so the lower the humidity, the higher the rate of transpiration. Okay, so these are the second one. So okay, for the temperature, the higher the temperature, the higher the rate of transpiration. Okay, I just give you the what you call the theory. Okay, no need to go through this because this one is just a waste of time. Then for movement of water, movement of air, sorry, the faster the air movement, the higher the rate of transpiration. So you see, okay, so now So in short, okay, this is the rate of transpiration. I mean the under the problem. So still air means no air movement. So the rate of transpiration will decrease. Okay. Wait. Okay. Your under a damp area means the humidity increase. So the rate of transpiration decrease. High temperature, the rate of transpiration increase. Okay, low light intensity, the rate of transpiration decrease. So in short, let's see whether they got. Yeah, later on we will show here. So you see, okay. Now, in order to reduce the excessive, we know that you see, plants usually have fewer stomata on top surface. Okay, of the leaves compared to the bottom surface of the leaf. Why? This is to reduce the excessive water loss from transpiration, because top surface of leaves are more exposed to sunlight. So when more exposed to sunlight, the temperature on the top surface will be higher. When higher, evaporation rate become faster, so the transpiration rate will become faster as well. You understand? Okay. So now the plant lost the water through transpiration. Okay, and we know that the stomata play as an important role in this process. So how the plant, okay, the plant that lives in desert adapt, okay, with the condition of air scarcity. So what they do, because the water is less, so the stomata remain closed during daytime, okay, and only open at night time. At night, so that it can prevent the loss of water. Okay. All right. So in short, the rate of transpiration increase here is increase when the light intensity is high, humidity low, temperature high, and there's a air movement. Okay, so this is how. Huh? Hey, where's my... Okay, next. Let's see the transport system in plant. Okay. So let's see the transport system in plant. Okay, you can see this is the decotyledon plant and monocotyledon plant. This is how the structure inside. Normally, inner one will be the inside one. The one in the center normally will be xylem. The one at the outside will be phloem. Same goes here. Inner part, inner ring is xylem. Outer ring will be phloem. And then same goes here. This one is xylem. This one is phloem. Okay. And this one for the monocotyledon, okay, the round part will be phloem. The one with sharp edges is the xylem. Okay. The cross section of their stem, okay, inner part, the one that more like the sharp, more to the sharp part is the sharp half, is the xylem. The round part will be the round half will be the phloem. And in the leaves are almost similar. Top part will be xylem, bottom part will be phloem. So this is how the structures of the what you call structures of the what you call that the plant looks like. Sorry. Okay, I think you better jot down this one. Okay, because I think your textbook never shows it didn't shows the monocotyledon and the what you call and the dicotyledon one. I think he only shows. Yeah, you need to draw as well. No need to draw like always Don't draw. No need to draw so detailed Okay. Okay, draw it first, and then let me know. Okay, then let me know. Okay. All right. So let's see the. So okay. So the structures that involve. Okay, the structures that involve in the transport system of plant will be xylem and phloem. Okay. So now the shape of this structure is their tube shape, tube. So the location of this structure, okay, in dicotyledon plant, xylem and phloem are arranged in a ring. Phloem is at the outer side of the cambium. For monocotyledonous plant, the xylem and phloem are scattered. Sorry, not arranged. Okay, they are scattered. You see, the one that you draw outside, you see they are scattered around. Okay, xylem has lignin wall. Phloem has cellulose wall. Okay, why? Because lignin is waterproof. Xylem need to carry water, so he need a waterproof wall. While phloem is carrying the organic substance, so he need a stronger, how to say, a strong but hollow structure. So he use the cellulose as the what you call. The cellulose is some sort like a fiber, so to hold it. Okay. So xylem made up of dead cell, while phloem are living cell. Okay. Now you can see, so phloem, okay, he carry the dissolved food from leaves to other part of the plant. Xylem carries water and dissolved mineral from the root to other parts of the plant. Okay, so what happened if to the plant without xylem and phloem tubes? Okay, so you see, so without xylem, water and mineral salt, okay, cannot be transported. So your rate of photosynthesis will decrease. While without phloem, food that produce, okay, during the photosynthesis cannot be transported to the other part of the plant. So in the end, the plant will wilt and eventually die. Okay? Can I? You understand? All right, now let's see. Yeah. So let's check with, wait. Let's check with the transportation of water in plants. Okay, so this one we already go through, so I'll just skip. So the tissue, okay. You see, when with this experiment, we use the water with red food dye so that the water will be in red color. Then after we put it some time, after we cut the stem, what will happen that you will see some part become red color. Okay, so those red color part, the stented tissue, that observed in the slides will be your xylem because you carry the red color water. Okay, so xylem tissue they are found in roots, stem, and leaves of the plant. So why the stem and leaf are also standard red? Because the color water absorbed by the root transport by xylem up to the stem and to the leaf. So that's why the xylem part all will be in red color. Okay, so the pathway, okay, pathway of water transport will be root hair to root to stem and to leaves. So the functions of xylem is to transport the water and mineral salt, like we say, from root to leaves, so that to enable the green plant to carry out photosynthesis. Okay. So now, while for the food, okay, you see, we remove the ring here. Okay, if you want to see, actually, we won't remove like this. Normally, we remove at the vertical part. So, according to your textbook, you should remove like this. You see, you should remove the ring here. Okay, so now, so you should, after 2 weeks, you should look something like this. Okay, so what happened to the above the ring? So above the ring, part above the ring, what happened? So it's swollen, and there is phloem. Why? Okay, this is phloem. So why? Because phloem transports synthesis food from leaves to roots. So in the cut part, there is no phloem tissue because already been ringed out. It's been already took out. So the synthesis food substance will accumulate in the region above the ring. Okay, can you understand? All right, jot down this one as well. You jot down the drawing here. You jot down this drawing. Okay, this drawing here, this one. Okay, and then this one, right in the side. Why this swollen? Okay, can I? You understand? Okay, jot down this one and this one, this and this. Yes. Okay. All right. Jot down. Then let me know. The thing that I ask you to jot down, you must be careful. Means that somehow it will come out in your exam normally. Right? Cheating on the Malay first and second paper. Indonesian boy. Oh. Wow, cheating in exam is a what to say, it's a normal thing lah nowadays. Your teacher maybe they already found out, they already found out, but they just pretend they don't. There is a chance. Okay, done. Let me know. Then we continue. Oh, they didn't. Well, I think they noticed. It's just that he don't want to make it a havoc, so they just pretend they don't. So it's okay. So let's continue. So what happened to the part below the ring? Okay, you see, for the part below the ring, it will shrink. Why? Because the plant tissue do not get the food synthesis by the leaves. So that's why it shrink, because not enough food, no nutrient. So the inference, we know that inference, we said the foreign tissue are located on the outer side of the cambium, where xylem located at the inner side of the cambium. Okay? So this is how. All right, you jot this down as well. Okay, because just now we already go through the half part. So the other half is this one. Okay, right now this one. I mean only two only. The two only, three no need. Just two only. Okay? Can jot this down first. Oh, of course, yes. Mhm. Oh yeah, but okay, do the questions. Okay, do the questions. Okay, then let me know. Huh? I'm going to toilet for a while. If you are done, wake me for a while. Okay, I think it's still you do now. You are answer until which questions? Number 5. Okay, then you done. Let me know. Then we can discuss. Okay, all right. So let's see the answer. Okay, so for the first one, what's the meaning of transpiration? So transpiration is a process of loss of water in the form of water vapor, okay, from the surface of the plant to the air through the evaporation. Then second, we underline the correct answer. So loss of water from plant through the transpiration is in the forms of vapor, okay, while the loss of water through the exudation is in the forms of liquid. Okay, so the tissue that transport water in the plant is xylem, okay, while the tissue that transports sucrose is phloem. The sucrose is actually the product of photosynthesis. Okay, and then next, state the 4 factors that affect the what you call that rate of transpiration. So it's light intensity, air humidity, temperature, and air movement. Okay, next. Why we use the dye to investigate the direction of water in the xylem? Because this will shows the location of xylem. Because the passage of water in xylem can be detected with the use of dye. Somehow, right? Because the water is colorless, so that's why we can use dye as the what you call. We can use the dye for the what you call that color. Huh? All right, then the final questions. Wait, so this shows the structures of xylem. So now PQRS, you see, PQRSPU, he wants you to show which one is xylem, which one is phloem. So the answer shall be like this. Okay, PURSTU. So this should be the answer lah, where P, S, and U, P, S, and U are phloem. Others Q, R, and T are xylem. Okay, can I? All right. So I think I will stop here for today lah. So good luck with your exam. See you. Bye bye. Okay, bye bye.